Okay, so I'll carry on where I think I left off a week ago. As far as I remember, I was talking about the basics of quantitative genetics, about the idea that you can describe individuals by analyzing their phenotype into non-genetic and genetic components, and the genetic component into additive, dominance, and so on. But most important, one can divide the variance in the population into various different components, and one can estimate those by looking at resemblances between relatives. So that's a very general framework, which applies really to any genetic model. But to be useful, one has to be able to have some faith that the variance components will stay more or less constant, or at least be predictable. And that prediction, that predictability, comes from what we now call the infinitesimal model. And I introduced this as a model one can define at the phenotypic level by saying that if you take two parents which have particular breeding values, then the offspring of those parents will have a variance in breeding value, which is Gaussian, and crucially, which has a variance independent of the values of the parents. The mean will be at the midpoint, but the variance is simply some fixed quantity, which represents the variance hidden in the population in heterozygosity, and which is released by segregation of alleles in meiosis. Okay. So this is a really simple model. Really, the key is actually to realize that the variance released amongst offspring does change as a result of inbreeding. If the parents are related, if the genes that come together in meiosis are related, then that reduces the variance that's released. But we can predict what that is. And the key point is that the variance in the population, the variance components, are not perturbed appreciably by selection. And this works remarkably well. We'll come to some evidence for that in a minute. And in fact, the idea is usually thought of and introduced as a limit of a model with additive effects, where if you have a very large number of loci with very small effects, then in this limit, you get this very simple model. But one can extend it to include dominance and epistasis with certain restrictions. And the real idea is that if you take two individuals in a population, knowing their phenotypic values tells you very little about their precise genotype. So the infinitesimal model really relies on the idea that there's a huge diversity, and I think the term redundancy has been used, but say that, a huge diversity in genotypes which produce essentially the same phenotype, and that the range of phenotypes segregating in a population is actually much smaller than the possible range that could be produced by selection, which is what we observe. So that was the sort of background that I introduced last Tuesday. And what I'll do now is talk about limits to selection. And I'll start about talking about limits to how far the mean can change. If you have a population of a certain size and we just select on the standing variation, we ignore new mutation, etc., we just select and select, eventually we'll lose all the variability through inbreeding. How far will we get? How far will the mean change? And this problem was solved in a really beautiful paper by Alan Robertson in 1960, which also gives us a very nice connection between the infinitesimal model at the phenotypic level, the quantitative genetic model, and the underlying behavior of the alleles. And so it's a connection between, if you like, population genetics and quantitative genetics. And I'll talk about how this extends to include epistasis. I'll make a slight diversion to look at what happens when we go beyond the infinitesimal and ask, with very, very strong selection, when the infinitesimal breaks down, then how far can we push the mean? So all of this is about limits to selection on the mean under directional selection. And then I'll go on to think more generally about limits to what selection can do. It's extraordinary how elaborate the molecular machines are that have evolved, the behavioral adaptations, the developmental programs, etc., all the things that fascinate us about biology. How does selection, this law of the higgledy-piggledy, as it's called by Herschel, how does that achieve all this? And what are the limits to what it can achieve? What is the limiting factor to the power of selection? And I'll just sort of talk about that in rather general terms. And show that the limiting factor is the product of population size and heritable variance in fitness, a quantity that's come up several times already. And then end by again mentioning something that we've already talked about actually in the previous talk by Michael Lynch, and I think in several talks before, this idea that we can estimate the variance in fitness, the heritable variance in fitness, from the pattern of correlations of allele frequencies through time. Okay, so that's a kind of overview of what I'll try and cover before we get all too tired and hungry. Okay. I should say that what I'm showing here is a sort of slimmed down version of notes that I'll put on the Slack channel. There's, I think, a version of the notes up to today's lecture on Slack, and I'll put an updated version with tomorrow's lecture notes on Slack. Also, I'll distribute answers to the exercises that some of the students came to last Friday. And in fact, we sort of were leading into what I'll talk about now in those exercises. Okay. So we start by asking, about the change in the mean. And in one generation, that's simply the selection gradient times the additive variance. And we had last time that the additive variance is just a sum over all the loci of the square of the effect times the product of the allele frequency. So a very simple formula for the additive variance in diploids. And let's say that's the initial variance, and we ask, how does that change as we inbreed a population? Well, the heterozygosity simply decreases by a factor 1 minus 1 over 2ne per generation. So after t generations, the heterozygosity at each locus has decreased by this factor simply through inbreeding. And a crucial simplification is that the additive variance depends simply on a sum of heterozygosities, and the only change is that each heterozygosity, whether p is small or p is large, whatever value of the allele frequency, they all decrease by the same factor. And therefore, that means that we could rewrite the additive variance like this. And in fact, we could have derived that directly from the infinitesimal model. Enrique, is that a question or an adjustment of the glasses? Okay, fine, fine. Yes, I shouldn't, yeah, anyway. So actually, I went through that thinking about the allele frequencies, but we can also go through it and derive directly from the infinitesimal model the fact that in any generation, the variance released into the population is decreased by a factor 1 minus f, and therefore we get this reduction in additive variance through time. Okay, so then we actually have a really simple formula for the change in mean over generation t is just proportional to the additive variance over t. And what we really want is something that's called sometimes R infinity, the total response, the total change in mean, or we could write it as sum of the change in mean over time 0, 1, 2 up to infinity, which is equal to beta times the sum of this thing, which is going to be BA 0 times the sum of okay, so we've simply got an additive variance that's decreasing geometrically over a time scale of 2ne generations, and that simply sums to keep this in. So this selection gradient times the additive variance in the first generation is just the change in mean in the first generation. And what this is saying is that because the additive variance is decreasing over a time scale of 2ne generations, the total change summed until we've run out of variation, everything's stopped, that's just 2ne times the initial change. Okay, so really, really simple formula. And in fact, that's sort of almost trivial. It probably was well known before Robertson wrote his 1960 paper. What he was doing was making a connection with the relatively recent sort of population genetics being developed by Kimura and others, to think about it in terms of the underlying allele frequencies. So what Robertson did was say, well, instead of doing all this sort of sum over all the generations, let's just think about what the ultimate expected change in the mean is. So we call it either R infinity, or we could call it, let's just call it that. I'm in danger of having a proliferation of notation, but if we think of the underlying alleles, then the change over the whole process is, call it delta star, is the change in allele frequency from the beginning to the end. That's kind of trivial. But the expected change in allele frequency is really just the chance that the allele fixes in the population minus the chance, minus its initial value. So what's happening is that we imagine a population with a whole set of alleles, some of them rare, some of them common, some distribution of allele frequencies. And in the long run, they must either be lost or fixed, okay? So we're running this whole process so that alleles that start at frequency P0 have some probability U of fixing 1 minus U of being lost, okay? So the expected frequency at the end is just U. It's the chance that they're there rather than having been lost. And so we can write the total change as this, where this is the probability of fixation of the allele, and this is its initial frequency. So it's kind of very, very simple, but it was a key insight to solving the problem. And then we can see that if there's no selection, then the chance that an allele at frequency P0 will be fixed is just P0, because basically you've got a whole lot of genes in the population, 2n genes in the population. A proportion P0 of them are of one type, 1 minus P0 of the other type. Now, one of those genes is going to be the ancestor of all the genes in the final population. So the chance that that ancestor is of a particular type is just P0 if there's no selection. So we're really looking at the bias introduced by selection increasing the fixation probability above P0. And that's given by this simple formula, which is usually attributed to Kimura. I'm not sure if it goes back further than that, but it's kind of a neat formula that is this form. So if we look at this, you can see that if P0 is 0, right, if the allele isn't there, then e to the 0 is 1, so this is 0. So fixation probability is 0, so that's fine. If P0 is 1, then it's something or other divided by something or other. That's 1, so that works out. And another kind of check is to say, well, suppose selection is very weak, so suppose 4NES is very small, then you can do a Taylor series. You can say that 1 minus e to the minus x is approximately x plus terms order x squared. So if selection is very weak, then you have basically this over this, which is P0. So everything sort of makes sense. And in fact, we can plot the graph of NES along here, 0, 1, 2, whatever, very accurately. That's just a line to calibrate. So, this is drawn properly here, but here's U, going from 0 to 1. Basically, if selection is 0, it's got to go through P0. So if P0 is, let's say, 0.3, it'll go through there. If it's got very negative selection, it's very deleterious, it's got no chance of fixing. If it's very advantageous, it's almost certain to fix. And so we have a sort of curve like that. And then if we had an allele that started at a higher frequency, say here, we'd have a curve that sort of looked like that, right? So we have a sort of sigmoid curve. So it's kind of a complicated thing, and then you wonder what to do with it. Well, what Robertson saw is that, well, you can make an approximation by saying that we'll look at the slope of this for weak selection. So the key approximation was to say, well, suppose selection on these underlying alleles is weak, so that selection is really perturbing the fixation probability a little bit away from P0. Then do some calculus, and I won't go through it. Well, it's pretty easy to do, so if you like calculus. So, yeah. So you can ask a computer, or your smartphone probably now does it, I don't know. But yeah, you do a Taylor series, you keep second order terms and so on, and you find that U is approximately equal to P0, the initial thing, plus 2NES P0 Q0 plus terms order NES squared, okay? You're telling him the answer to that. Anyway, okay, so I'm sort of going through this, in the way that Robertson went through it, and if you then plug that in, this formula, let's just write it out. Yep. This bit. This bit. Ah, sorry, I wasn't writing very legibly. Okay. I just meant of order, O means of order, NES squared. And all that means is that I'm assuming NES is small, 0.1. So the extra terms are of order, 1%, 0.01 squared. And we can ignore them. So that's just sort of a mathematical way of writing this Taylor expansion. So we're just, geometrically what we're doing is saying, let's take the point at the origin. We'll say that NS is small. And so we'll just look a little way away from there. So we're taking a straight line, which is a tangent to that line. And the slope of that is 2NES PQ. So then if we put that in here, so we have this formula. That's just the sum of 2 alpha I times 2NES P0 Q0. Now what's S? Well, we have a selection gradient beta, we have an allelic effect alpha I, and so we imagine the selection gradient is simply defined as the increase in relative fitness for each increase in trait value. And so S is just alpha times beta, so we can take that out there. And so this is then the sum of 2 alpha I squared beta times 2NE times P0 Q0, which looks a bit messy, but we can just reorder the things, and we end up with 2NE beta times sum of 2 alpha squared P0 I Q0 I. I should have written the little i's there, because this is a sum over all the different alleles in the initial population, all the different loci, which might have different allele frequencies initially. But that is just equal to the initial genetic variance. Okay, so it's another of these quantitative genetic miracles where everything cancels out and all the genetic details disappear, and we get back to this result which we had by a very simple quantitative genetic argument that didn't really require us to know about Mendelian genetics. We just had to have the infinitesimal model. But what Robertson was doing was saying, well, actually, we can understand this in terms of fixation probabilities of a whole lot of alleles underlying them. And what's crucial, at least I think is crucial here, is that this shows that the underlying assumption of the infinitesimal model is that the change in mean is the cumulative effect of many slight perturbations, right? So it's a perturbation away from neutrality. And I think even at this august meeting, I've heard heretical remarks and statements that the infinitesimal model, polygenic adaptation generally, involves slight changes of allele frequency. Well, it's not really so. In this model, we've got big changes of allele frequency. We've got fixations and loss. And so it's not that the allele frequencies don't change much. It's that they have a distribution which is very close to what they would have in the absence of selection. So selection is perturbing lots and lots of alleles. And I find this a very wonderful and beautiful theory. For many people, it may be deeply depressing, because it would mean, if we were in this regime, that you couldn't detect selection at individual loci, that they would look almost neutral, right? So, I think it's quite a big question, what fraction of adaptation is truly polygenic in the sense that it's close to neutral. And it's, of course, we've successfully sequenced lots of genomes and detected lots of loci that are under selection and that contribute to adaptation. And the problem is, what fraction of the adaptation does that account for? So I'll leave that there. Any questions?
So just a quick comment. One other thing that is more for people to think about breeding is if you select a population by truncation on a trait, the stronger your selection, the larger beta is, but the smaller your effective population size.
Yes.
So there's a trade-off, and Robertson found that the optimal selection to get the largest long-term response, trading off NE and beta being inversely related, is to select half the population each generation, which is very counterintuitive.
Yeah. And in fact, he and other people at the time, there's a whole literature then, which is quite elaborate, which asks, what is the optimal truncation point if you're allowed to vary it through time? It's not necessarily just 50%. There's a rather complicated solution which does a little bit better. And I actually rather foolishly got a grant to work on the relation between evolutionary computation, these genetic algorithms that computer scientists who can't be bothered coding and thinking, or they just do natural selection, right? Select amongst algorithms, see what works. They have conferences, a couple of conferences a year with little theory sections, and they try and develop theory. But they hadn't any idea that one could write down this sort of argument, because it actually applies to optimizing the algorithms. What fraction do you select? They have somehow stumbled on the idea that you need recombination, you need mutation, you need some kind of selection, truncation selection works best, and they have different terminology. But it's interesting, this was all out there in the 50s, really, 60s. Okay, so what we've got is, we start with a population with a whole lot of polymorphic alleles, which have slight effects on the trait, right? And we are asking, how far can we shift the mean by just selecting for a long time, right? We're allowed to select for as long as we like, and eventually we're going to lose variation. So the point is that the change in mean will be proportional to the difference between the fixation probability, the chance you fix the allele, and its initial frequency, right? So if it's neutral, its chance of fixation is the initial frequency, and there's no shift in the mean. Some go up, some go down, but on average, there's no change. So you're trying to bias by selection. What selection is doing is biasing the fixation probability. So it's the extent to which it can be biased up above that P0, which in there is 0.2 or 0.5 or 0.8. Yes, that's right. So it's an expansion really in NES small, and therefore U minus P is given by this thing, 2NES times P0 Q0, which is a small value. Yeah. Okay, so that's the basic theory, and actually there's a really nice paper by Weber and Diggins, which actually one of the examples in the exercise was based on. So a genetics paper, in which the main advance in this paper was the invention of the inebriometer for determining how resistant to ethanol flies were. It was a way of selecting on very, very large populations. And in the discussion of this paper, Weber and Diggins sort of asked, well, if we make a meta-analysis, survey, I don't know, 20 or 30 different selection experiments that have been done on maize and mice and flies, a variety, mostly maize, mice, and flies more than anything. And what they did was plot the change, not the change after an infinite time, but the change after 50 generations, which most of these experiments lasted of that order. That was about the limit of human patience. So the ratio, change in the mean over 50 generations minus the change in the first generation, the maximum really is 50, right? So if you had an infinitely large population and you just kept going at the same rate with no slowdown, you get to 50, right? And that's the right-hand side here. So what we're plotting against is effective population size estimated from the number of flies that were counted and so on. And the exponential curve labeled 2NE is Robertson's limit. This is plotted on a log scale. So Robertson's limit would be where you'd get to after an indefinite time, after a very long time. That would be this 2NE. The two curves to the right, the lower one is the theoretical curve, which is basically this formula summing not to infinity, but summing over 50 generations. So it's 1 plus 1 minus 1 over 2n plus 1 minus 1 over 2n squared. That's an easy sum. And the curve that's slightly above it is throwing in Vm of 10 to the minus 3, which was known to be about the right Vm. So a little bit of mutation doesn't really make very much difference over this time scale. So all of that says that we expect from this infinitesimal argument that after 50 generations, we get to this curve, the lower curve there. And the points don't fit that curve, but they're not far off. They're following it reasonably well. And maybe the difference, they're slightly below it because selection is reducing variation, because actually we're not in the infinitesimal limit and selection is weeding out major alleles. It could also be linkage, which will have a similar effect. Linkage will account for much of that. So I'm really struck that you throw in data from a whole lot of rather hokey experiments on all kinds of different organisms, different traits, and you get something that reasonably fits this. And that suggests that over this time scale, the infinitesimal model will work pretty well. And of course, that's the experience in artificial selection, that over timescales of 50 generations, 100 generations, things work pretty well. You can basically predict what will happen just from the effects of inbreeding. Sorry, I should use this elaborate technology. Okay, this upper curve here is Robertson's limit, which is for an infinite time. R infinity is equal to 2NE times the initial change. So this is just, it looks like a curve, but we have a log scale. If this was a linear scale, it would be a straight line. Okay, but we're not going for an infinite time, we're going for 50 generations. So we have something that looks like that. We can calculate that more or less in the same way. The lower curve is the simple prediction summing 1 minus 1 over 2n to the t for 50 generations. And the one slightly above is throwing in a bit of mutational variance. Because a bit of mutational variance. So what we talked about, I think, last time is that, and I think Michael Lynch talked about, the VM is about 0.001 VE, maybe a bit more. That's fairly consistent. So if you put in your guess at how much mutation might be contributing, you find it doesn't make very much difference over 50 generations. And this is actually, I think, paper. Were you on this paper with Bill Hill a long time ago? Trying to remember. So anyway, Bill Hill had a paper on the contribution of mutation to selection response, basically making this argument. Okay. So yeah, so it's not a perfect fit, but it's doing pretty well for biology. I think that's a pretty good fit if you squint at it from a distance. So that suggests that it's not unreasonable to think that at least over short terms, 100 generation timescales, the alleles we're looking at are not strongly selected relative to drift. So drift and selection working together. So I'm going to spend a bit of time. This is getting a bit more complicated, but just, yeah. Sorry, what? When would the infinitesimal model break down? Yeah. Well, I mean, the obvious difference is when you have, fewer loci with larger effects. So yes. How many would you need? What would be the fraction? Okay. Yes. Yes. Not try. Yes, yeah, I'll come to that in a bit, actually. Yes. Generally, it'll increase the loss of variance because you're actually having selection forcing things to fixation faster than drift would. So this whole calculation is about drift leading to loss of variation. If you have selection also leading to loss of variation, that speeds things up. And I suppose one way of getting an idea of the timescale is that if you have an allelic effect alpha and a selection gradient beta, right, and we can work that out from if you have a truncation selection or any selection scheme, so your selection gradient, the selection coefficient is beta times alpha, And the timescale of selection is of order 1 over alpha beta, right? So if an allele starts out common, it's going to be getting near fixation or to high frequency in of order 1 over alpha beta generations, which will be shorter than the drift timescale. Now you can say if you've got a really big population, you could have really rare alleles present in 10 copies, and they take a long time to get to higher frequency of order log selection, right? So you can extend things, but the log doesn't get you very far. It's still going to be of order 1 over alpha beta. So that will give you some idea of how long you have. I think in the sort of Weber and Diggins kind of lab experiments with smallish population sizes, go back there. I mean, most of these experiments were done in the 10 to 100 NE, that's these. And these ones were all Ken Weber with his fancy machines. He did various experiments doing bigger ones. Once you're out here, then of course, weaker selection can still be significant. But in here, you need a pretty strong selection coefficient, pretty large effect to be significant relative to just plain drift. Yeah. Okay, so the reason I'm going to talk probably a bit too long about epistasis is just that there's a lot of talk in quantitative genetics, in population genetics, about the importance of epistasis. And of course, there's also the general feeling, and I think I somehow feel that more mechanistic molecular biologists poke fun at quantitative geneticists for working with an additive model. It's obviously stupid to think that we're the sum of the effects of our genes. It's kind of quite difficult to get your head around the idea that the additive model does give good predictions, as we just saw. And so people say, well, you've got to include epistasis, and epistasis will change everything. And in particular, there's an idea that epistatic variants can be converted into additive variances. Conversion sounds a bit mystical, really, but it's a very simple idea. Suppose you have a situation where the marginal effects of alleles are small. So it doesn't matter whether you have this allele or that allele, it doesn't affect the trait. However, if those alleles are interacting epistatically with other alleles in the population, with other loci, then as allele frequencies change at those other loci, the marginal effect of the focal allele will change. The whole point about epistasis is that it says that the effect of an allele depends on the genetic background. And in fact, this is something that can be predicted within the framework of classical quantitative genetics. Because you can show that if you divide up the variance in a population, let's think of a haploid population to keep it simple. You can take any population with any arbitrary mapping of genotype to phenotype, the arbitrary fitness landscape, and define what the additive variance is, what the additive by additive variance is, what the third order interaction is, etc. And the additive variance is simply the variance of the marginal effect of the alleles. And you could come up, you had to work quite hard, but you could rig a model such that that was zero, but there were additive by additive interactions. The trouble is, as soon as you perturb the allele frequencies, the marginal effects will be generated by the change in allele frequencies. And that is a predictable effect. And so there's a formula here which says that the expected additive variance after a certain amount of inbreeding is equal to this formula. Well, the additive variance initially is dissipated by a factor 1 minus f. We saw that already. That's the reduction in the release of variance due to inbreeding. But if we have additive by additive variance in the population, then you generate additive variance at a rate 2f 1 minus f. And if you have additive, typo, it's a mutation. It's been shown actually that the rate of generation of typographical mutations is always 10 to the minus 3 times the size of the text. Something in that order. Yeah. Yeah. So, okay. So let's draw a picture of that. So we put here additive variance at some time after there's been a certain amount of inbreeding, and here's the amount of inbreeding here. There's going to be a component that comes from BA 0 times 1 minus f. That's the most basic formula, really. But then the epistatic components of variance will contribute some, VAA at time 0 times 1 minus f times 2f, I think it is. And then there'll be a component from and so on. You sort of carry on with this. And one way to think about what's happening is that what drift and inbreeding are doing is forcing allele frequencies from intermediate values towards fixation, towards being very low or very high. And when alleles are rare, when they're close to the boundaries, then the variance they produce has to be mostly additive. Because if you have an allele coming in, it won't see many interactions with other alleles. It'll be mainly having a simple marginal effect, because the different alleles are all rather rare, rather close to fixation anyway. So at any rate, you have this rather general formula, which gives us something that looks rather intractable. But all we have to do is then sum over all the generations and say that the total change in mean, or the expected value, it will obviously vary from replica to replica, but the expected change is the selection gradient times the sum of the expected additive variance in each generation, which starts out at VA0 and then is given by this formula. So we now have a double sum, and it kind of looks even worse, but actually, again, by a miracle, it all cancels out, and it all resolves. And in this case, for haploids, you get the amazing result that it's almost the same as Robertson's result. The only difference is that we've got beta times NE times the genotypic variance, which is the sum of VA plus VAA plus VAA. So it's larger. You do get further. There is a conversion of non-additive variance into additive variance, but it's constrained by the total being given by the genotypic variance. Okay? So notice there isn't a factor of 2 here, which we had in Robertson's result, because we're doing haploids. So it comes out rather simply with haploids. If we do it for diploids, we actually get something very similar, but we actually get a factor of 2 to the k minus 1, amplifying the effect of the non-additive variance. So the non-additive variance does contribute relatively more in diploids. And you could imagine this could blow up, but it's pretty hard to see how one can do that. One can concoct really bizarre models that will do that. But in general, what happens is that the sort of kth order interaction effect looks something like epsilon k, which is some epistatic coefficient for the interaction between k different genes, times pq at the first locus, times pq at the second locus. Let's say this is the third order one. It's going to be a product of 3 different heterozygosities. And these pqs can't be greater than a quarter, right? That's the maximum. So even if all the loci are highly polymorphic, are near the middle, which is the best possible case, you can't really get the higher ordered additive terms to increase enough to contribute. So they're decaying faster than this is boosting it. So that's a bit of a hand-waving argument. But we do have a sort of a concrete result, which says that if you have an arbitrary model with epistasis, and you just ask what's the change in mean under continued directional selection with constant selection gradient, it's limited by the variance components. And so we know what the variance components are. We know that they are mostly additive with a bit of second order. It's very hard to measure the higher order one, so we can't exclude the possibility that there's sort of magic higher order epistasis that will contribute to the response, but it seems pretty unlikely. Okay. Any questions? Yeah.
Can you explain the 2, 3?
Why is 2, 3?
Yeah, I didn't quite understand that.
I don't. I would have to look it up in Lynch and Walsh, I think.
It's not intuitive.
I mean, I think I could probably, given a day or so, come up with the derivation. But I mean, yeah, where it comes from is thinking actually about probabilities of identity amongst multiple sets of genes. And then you do need to make some assumptions to relate those to the sort of standard inbreeding, assuming it's a simple sort of Wright-Fisher model and so on. So it gets quite intricate and so on, but it's kind of, given some reasonable assumptions, I mean, assuming it's a fairly well-mixed population with a simple inbreeding, then this works. thought Bruce would say it's page 1333. It's somewhere in there. Sorry? Yeah, it goes back to the old classic animal breeding in the sort of 50s or something, 60s. Anyway, I'll take a diversion actually, because this is just to say that you can get somewhere even without being in the extreme infinitesimal world. So all of these arguments which use sort of classical QG are assuming that NS is small, so drift is really what's causing the loss of variation. But suppose we ask what happens if you have really strong selection, so that you have a really, really big, enormous population, and moderately strong selection, so that NS is very large, and we can treat the whole thing really deterministically. And the picture one can paint is actually sort of easiest to see on a log scale, where we plot log P over Q, the log of the ratio of allele frequencies that we've seen a few times before. So this is 0.5 here, and this might be 0.9, and 0.1, and 0.01, and 0.001, 0.0001, but it's a really big population, so we can get down there, you know, population of 10 to the 8 or something. So we go up here, 0.9999, something like that, whatever. And then you have this result due to Haldane originally, which is under a constant selection, which in our case will depend on the allelic effect. But if you had something that was moderately selected, it would just change in a straight line. So that actually it'll spend most of its time going from very low frequency up and up and up, and it takes the same time to go through each order of magnitude, if you like. Same time to go from there to there and there to there, more or less. And you'll only actually see it when it passes through, well, no, I should have drawn it like that. Very good at drawing these things. Let's say yes. There's a kind of relatively narrow window in which what you see on a normal scale would be a sudden sweep from low frequency to high frequency. So that actually gives you a way of understanding how epistasis is going to affect things. Because suppose you've got a whole lot of these things, and they're all starting at some initial rate, and some of them are just going to go down, and we don't care about them. And some of them are up here, and they're going to go up. We don't care about those either. They care about the ones that start rare, and they're going to go common. And they're going to contribute an amount equal to their allelic effect. But the allelic effect now is being influenced by epistasis, so it depends on the background. So what we can say is, suppose we consider this one. This is the first one to sweep through. Then another one's coming along, and suddenly its background has changed. And so it might have a slight, I don't know, we want it to go up or down? Vote. No one cares. It's going to go up, okay. But now, this one might change, it might slow down, but it's already common, so it doesn't really matter. And then you could have, I don't know, you can keep going like this, you can keep drawing pictures, this one's coming along and it comes to here and it actually gets deleterious and then comes to here and then it gets an advantage, right? So you have this picture of successive sweeps, and each time there's a sweep from low frequency to high frequency, you go through this window, you change randomly the selective value of the others. And of course, if you have an arbitrary model, it really can't say anything in general, but what I looked at, this is a paper with Thiago Paixão, who's now the Gulbenkian. What we did was just assume there was some random pairwise epistasis, and that's kind of a neat model. You just say, let's throw down some random pairwise coefficients, so that if you have a change at one locus, it causes actually a randomly distributed change in selection coefficient at the other. And what you can show is that if this isn't systematic, if it's just random, there's no change in the net response unless you end up somewhere different from where you would have done before. So if you end up at a different adaptive peak as a result, that is, if something that would have fixed actually is eliminated or vice versa, then you can actually increase the expected response. And you can estimate, you actually, surprisingly, to both of us, I think you could get a quite simple formula for the expected increase in response provided by epistasis. So then, we got excited about this and we did some simulations. And this is actually a summary, something like the Weber and Diggins curve, actually. Log scale of NS now, so effective population size times selection gradient, scale relative standard deviation. And this is the ultimate response, the net change divided by standard deviation, genetic standard deviation, or log scale. So we have this Robertson limit here. This is in the small NS regime. It works pretty well up to even NS 2 or something like that. And then the top pair of lines is 1,000 loci. The bottom one is 50 loci. So we're getting, the more you get, the longer you stay in the infinitesimal regime. And then the black dots are an additive model. And is that right? No, it says here. No, the additive model is in gray, that's right. And the black dots are with a bit of epistasis, actually with fairly strong epistasis with different standard deviations. We chose the standard deviation of the pairwise coefficients 0.5, 5 times the standard deviation of the additive effect. So you could argue about what to choose for parameters, but basically there is an increase due to epistasis, but it's not enormous, and you end up at a limit that you can predict. These dashed lines are the theoretical prediction for the ultimate response. And the theory was for this very strong selection limit for NE very large. So this is just to say that epistasis isn't magic. You can actually do some calculations and make some predictions, as long as you're prepared to make some assumptions, which actually probably rather hard to justify biologically, about the randomness of epistatic coefficients. Of course, if it's systematic, anything can happen. If you say that we'll make a scale transformation and that the trait is no longer the trait, but it's some additive trait to the power 10, then you can make things, you can generate anything you want. So I must say I don't have a good intuition for what kind of epistatic models are reasonable. There's a whole zoo of fitness landscape models, which I think are generally chosen for sort of tractability rather than any biological sort of justification, but one can argue about that anyway. Okay, so what I'll do now is turn to a different kind of metric of progress. I mean, it's a little bit, dubious, I think, talking about progress at all in evolutionary biology. One of the books I like most, actually, is a book by Michael Ruse, From Monad to Man, which describes the really deep roots that the idea of progressive evolution have, even amongst right-thinking people like all of us, even postmodern synthesis. And it's a nice book because it gives a history of the subject from before Darwin, through the Darwinian times, through to the modern, well, almost the modern day, and has interviews with people like John Maynard Smith and so on, and I think Ernst Mayr. So there's a few interviews in the back of the book showing that, it's really hard to shake off, or even to know whether we should completely shake off ideas of progress. I think, Mike, you're doing the best job in showing that actually we're just a ramshackle collection of molecular junk that has accumulated. Our ribosomes are encrusted with a great weight of molecular complication. But that's not the usual view, I think. At any rate, we've been talking before about a tangible economic sense of progress, increasing the mean milk yield, increasing the mean of some trait. That's very tangible. But when we think of evolution in the long scale, we think of evolution of complexity and so on. How do you measure that? And the first person really, I think, to think of it in terms of information was Kimura, actually in 1961, well before he was working on the neutral theory. He had the idea that whenever an amino acid was substituted one for another, that was putting in a number of bits of information. And he actually used the measure that Shannon had only a few years before defined as an information measure. And Kimura's measure was basically the log to the base 2, if we wanted it in bits, of 1 over the initial frequency. And the logic was that if the population was evolving neutrally, this allele would fix with a probability p0. So an allele's there, it would have some slight chance of fixing. That's probability is p0. If it's given a strong selective advantage, it's certain to be fixed. So this is just the log of the ratio between the chance it's fixed with selection versus the chance that it's fixed without selection. And it's a measure really of how far selection is concentrating the amino acid sequence around a particular best sequence. It's not directly a measure of kind of adaptation in the sense of mean fitness, actually producing more offspring, but there are all kinds of problems in using mean fitness as a measure of progress. For example, we can't all, mean fitness actually, mean absolute fitness cannot be much different from 1 in the long run, or 2 for sexual organisms. So this is a kind of measure of approach to or concentration around a particular set of presumably fit genotypes. And Kimura actually bounded this quantity by using Haldane's argument for substitution load, or what Haldane called cost of selection, which, various arguments which one can criticize in all sorts of ways, and Haldane thought, well, one couldn't have more than 300, more than one substitution per 30 generations, I think, for mammals, something of that order, based on the idea of the reproductive excess you need in order to substitute one allele for another. So Kimura made this sort of back-of-envelope calculation and said that one could accumulate 10 to the 8 bits of information since the Cambrian. So that was quite a definite thing, which maybe isn't, that's miraculously, it's probably our functional genome is of that order. So yeah, don't know what to make of that. So anyway, what I'll talk about is a result by Michał Hledik, who's a student who just finished with Kashper Tacik and myself, who came up with a very general bound for the rate of accumulation of information in this way, and generalizing to a thing called D, which is the log of the probability of observing a population, which we might think of a set of allele frequencies, perhaps, or a set of genotype frequencies, actually quite a general result, divided by the probability of observing that configuration without selection. So it's a generalization of Kimura's idea, really. It's the log ratio of the probability with selection and probability without. And this is called the Kullback-Leibler distance. It's a distance between 2 probability distributions. And so just to sort of illustrate what the idea is, you imagine the very simplest case of just a series of mutations thrown into a population. They have NES equals 2, so they're moderately weakly selected. If you have no selection, you get mostly loss, some fixations. If you have selection, you get a shift in which the plus alleles tend to become more common. So you can see the distribution over 500 generations pushing towards the plus. And of course, we have the whole diffusion theory developed by Kimura, which tells us how this works. And then you can just measure this Kullback-Leibler distance between the selected distribution and the neutral distribution. And this is a rather pathetic increase in information of about 0.8 bits, because you've sort of pushed the frequencies to not quite fixation, but to high frequencies. So you're the order of 1 bit. And you can do these calculations for all sorts of other models. So what Michał showed is that there's actually a bound on the rate of accumulation of information, which in the limit in Kimura's example would be actually the rate of substitution of alleles, but more generally it would also apply to infinitesimal model kind of scenarios where you have perturbations from neutrality at lots of loci. And this bound is basically a constant times the population size times the variance in relative fitness, or in fact this should be the heritable variance in relative fitness. Which kind of makes sense, because the information ultimately is coming from living or dying. It's the number of individuals who are either reproducing and leaving offspring or not. And the amount of information depends on how variable the number of offspring they leave is. So we have this sort of natural combination. And then you can actually ask, what would be the most efficient way to accumulate information? And for this particular example of just directional selection on mutations thrown into the population at some rate, that turns out to be the infinitesimal limit. And there's an intuition there which is quite simple. If you have, you might say, well, we just apply strong selection. So we have mutation comes in, we want to fix that allele, right? So we just select on it really strongly. But that requires a high variance in fitness. You've got to have individuals producing, a lot of offspring for some time, so there's a certain variability. There's a limit to how much you can do that, how many loci you can apply that to. So you can say, well, we can get the same outcome by having a weaker selection. We still have a very high fixation probability. This fixation probability formula asymptoted close to 1, provided NS is sufficiently large. So then you say, well, let's make NS weaker and weaker. And then you get down to a regime where NS is now small, let's say its NS is 1 or 2, so you're not certain to fix it. But you've got enough fitness variance, if you like, in your budget to start doing this at lots of loci. So it turns out if you ask what is the rate of accumulation of information summed over all the loci, it's maximized when you have a lot of loci, each under weak selection, exactly in the sort of Robertson regime, where you're just perturbing the fixation probabilities. So that's kind of intriguing idea. We haven't, managed to generalize that to make a statement that if we were divinely designed, we would be in the infinitesimal world. I'm not sure that's true, but it's kind of intriguing that there's this connection between the infinitesimal model, which is the way we actually describe quantitative trait variation, and this sort of optimal rate of information accumulation. Yeah.
Hello. Sorry to do this, but I got lost a while ago.
Oh, okay. How far at the beginning?
So how are we? I feel like we haven't enough time that I'm going to do this with you, but can you go back up a bit? First, how are we? I know you said this, but how exactly did Kimura define information? How are you measuring this?
So he was defining it in terms of a sort of information transmission. I can't remember the details, but he was taking it from Shannon, who actually, I think Shannon actually did a thesis in population genetics originally, and then switched to something possibly more useful. So anyway, so the example Kimura gave was to say, look, how much information do we gain if we establish a particular amino acid sequence rather than a random amino acid sequence? So if you just start out with completely random evolution, then you'll end up actually with, I'm sort of veering towards a mutation-based model, but it's easier to think of it in terms of a model where you start with, at each site, some frequency of different possible alleles, right? And let's say, I suppose it's a simple case, you have a population which has an equal frequency of A, T, G, and C, and then you ask, what's the chance that we can actually fix A? The probability with selection would be A, that's the one we've got. The probability without would be a quarter, right? So the neutral probability would be a quarter, and so then you have log to the base 2 of 4, so you've got 2 bits of information. So if you're just saying, how can we establish that we have a particular nucleotide rather than another, that's it. Kimura didn't quite make that argument. He was thinking not in terms of the DNA sequence, although of course DNA had just been discovered, so we all know, but he was thinking rather in terms of what he worked on, which is diffusion theory. He said, suppose we start with an allele at frequency P0, and what selection is trying to do, if you like, is establish that allele, but it's very rare. And so the information we gain is the log of the ratio between the probability of fixing it with selection, which is 1, divided by the probability without selection, which is P0. So if it's very rare, if it's 10 to the minus 6, then it's log to the base 2 of 10 to the 6, which is about 20 bits. So this is, and there's Chris Watkins, who's in London, is a computer scientist who has a series of papers which pushes this argument actually in the language of computer science. And his way of thinking about it is to say, well, and I think in these CS information theory things, they have Alice and Bob typically, and they have Princess Alice in a tower, and you can only communicate with Bob by selecting on populations of fruit flies and then sending in the populations. So the question is, how much information can you convey by selecting on a bottle of flies? This is a slightly weird way of looking at it, but okay. It's computer scientists, I'm saying. Anyway, yeah. Well, no, because the baseline is a neutral baseline. So really what you're doing is saying, what's the probability distribution under some regime with selection relative to the neutral? I suppose you could ask about information conveyed by mutation, but normally we would think of either the pure neutral thing, which Kimura was doing, or a mutation plus drift as the baseline, and then say we add selection. So you're comparing two scenarios, one with selection, one without selection. Yeah. The X is pointing. Okay, so what you're doing is just saying, well, you have a probability distribution for the neutral case, which is blue. That's the typical U-shaped distribution, right? And you have a probability distribution in red, which is under selection. and you take the ratio of those. So you take the ratio of the two, and then take the average of the log, and that's it. Yeah. Yeah, yeah. So it's sort of easier in the Kimura framework where it's either fixed or lost. But here you're, yeah. Laura. No, it's positive. So, I mean, you could be selecting against this allele and they all pile up there. You still gained information because you've pushed it to a particular outcome. There's nothing in here that tells you it's a good outcome, actually, which is a bit of a... Yes, yeah. So I think it's infinite, actually, because you could start at P0 is 0, right? And then it would be... Yes. I mean, I think the way Kimura started was just thinking about information sequence as a bit string, which is if we have 10 to the 8 functional bits or something like that, and that's 2 times 10 to the 8 because you've got 4 possibilities, so it's not very much. Your laptop has vastly more information than is generating you, which I find disturbing. Anyway. Okay, this was a rather abstract aside. I'll come back to something more concrete that actually connects with things that we've been hearing about, and I think I'll be finishing with this because I think it's probably time to be winding up. And this is just connecting with the previous section. We have this bound on how much information selection can accumulate. So then we think, well, what really matters is population size, fine, times heritable variance in fitness. But we don't know what the heritable variance in fitness is. And I started last week by saying, well, there are different views on this. So if you look from the quantitative genetic standpoint, you see all kinds of traits, including traits close to fitness, including fitness itself, seem to have a high heritability, or at least a high additive variance. And I think we had in some other lectures, sort of, remember, I think, was it Louis Miguel or Bruce, talking about how you scale fitness variance. But if we take the additive variance in absolute fitness, there are sort of hints, at least from long-term studies in the wild, that this could be quite high. And in fact, it has to be quite high if we're to explain why sex and recombination are maintained and so on. Why we can fight off pathogens, et cetera, et cetera. On the other hand, if you have sort of classical, molecular evolution type of viewpoint, where you think there's just deleterious mutation, which is limited by how much mutation load we can stand, by the observed rate, a total rate of deleterious mutation, then the additive fitness variance can't be more than 1%, 2% probably, even if deleterious mutations have quite strong effects. Similarly, substitutions are quite rare, in fact, per site, and so you can't get much additive variance. So we really don't know what the heritable variance in fitness is. So the idea that was taken up recently by Buffalo and Coop it came actually again from a really nice paper by Alan Robertson in 1961, again in the sort of animal breeding context, and something developed in some detail by Santiago and Caballero in 1995 in the context of linked selection, asking what is the rate of increase of allele frequency variance? And we have to be quite careful about defining this. I think it was, it appeared in Enrique's talk, so I'm really going to just elaborate on this a bit. We think of the change in allele frequency, defined as, let's say, pt minus pt minus 1 as the change over one generation. And we think of the variance of that as equal to p, well, all right, I'll do it like that, pt qt over 2ne, right? So that's our absolutely basic formula for drift. And that's the change in allele frequency over one generation. Now, if we have variance in fitness, that is actually what drives that change. You won't get any change of allele frequency from one generation to the next unless individuals have different numbers of offspring, or rather, unless genes have different numbers of offspring genes. But correlations from one generation to the next don't directly alter this quantity. What happens is that allele frequencies keep going up, so that if you have uncorrelated changes, the variance of delta P over T generations will go as T, right? A random walk has a constant increase in variance per generation. Whereas if you have something that keeps going in the same direction, then this actually increases initially as t squared until the correlations fall off, and then it falls away. So what we need to think about is the expectation of delta p, I'll call it, what do I call it there? Delta tp. So we're going to define delta tp as equal to p, call it tau, t plus tau minus pt. In other words, it's the change over tau generations. So we normally work with delta 1, but we could work with delta 2, 3, 4, and so on. And the expectation of that squared would, delta tau squared, will be greater than tau times the expectation of delta p1 squared. Because if things keep going in the same direction, you accumulate variance faster. And so what we can have as a measure of the accumulation of variance in allele frequency in the long term is this. In other words, we take a time slice of tau generations, and we take the variance of the change of allele frequency over that time slice, and then divide by the time slice. And of course, if there was just random uncorrelated drift, that would make no difference. But if there's correlated drift, this measure would actually increase and then level out. And it would level out at a time given by the correlation time span. You know, at some point, correlations stop. Of course, if you have a hard selective sweep, when things just keep going up forever, then this whole thing breaks down. So we're talking about a situation where I think you have a neutral allele sitting on a genetic background, and it's linked to stuff. It's on a background which is good one generation. Next generation will probably still be mostly on that good background, but it'll get passed on to different backgrounds over time, and it'll lose that correlation. And so what Santiago and Caballero showed, and what's used by Buffalo and Coop, is something like this formula. It's a bit of a nightmare getting the notation right, but the rate of increase of allele frequency variance, as defined there, is the standard PQ over 2n. And then there's three components. The half here for diploids, that half comes from segregation. Right? So even if every diploid individual has the same number of offspring, you'll have some variance in gene frequencies because at meiosis, one or the other gets passed on. This is assuming there's no meiotic drive segregation distortion. That could inflate that. Then you've got this component, which is the variance in diploid individual fitness, which if it's a Poisson number of offspring, it would be 2. So 2 over 4 is half. So this would be a half plus a half. So in the baseline Wright-Fisher model, you find you have PQ over 2n, with equal contributions coming from segregation and from variation in individual fitness of diploids. But then you have this other term where VA is the additive genetic variance in fitness, in diploid fitness, divided by something proportional to the square of the recombination rate. In fact, there's a slightly more complicated formula which allows for other ways in which the correlations decay. C is basically a measure of the rate of decay of correlations of fitness from one generation to the next. So this is really kind of a simple formula, which actually it's surprising that it took so long for people to realize that this is a neat way to estimate additive fitness variance without identifying individual loci. Okay? I mean, I guess it's only recently become possible because only now do we have really genome-wide data from many, many markers from time series. Right? And there still aren't that many data sets. But one of the best is Nader's data set. So Buffalo and Coop reanalyzed Nader's data. And so this diagram here, top one shows the covariance in units of 10 to the minus 3 of allele frequency change over successive generations. So if we start at one generation and go to the next one, that's the variance of change. And we go across 2 generations, delta P1 times delta P2, we get a lower value and we go down. This was, I think, is it 10 generation data set or 100 generations measured every 10? sorry, 60 generations. Yeah, that's right. So 10 up to 60. Sorry, that's right. So measured every 10 generations. Would have been good to measure them every generation, but won't go there. Okay. Anyway, so that's starting at a particular generation and then looking at the correlation over successive time points. And this is starting at Brown is starting at the next generation, and the next time measurement and correlation over successive time. So you're actually seeing correlations over 10, 20 generations. So quite a long correlation time. So that's the data from these 6 data points. And then you can estimate from that what they call G of T, which is the covariance. I'm trying to remember the definition now. Did I write? Sorry? Yeah, it's the proportion of allele frequency variance in the long run due to linked selection. So we have this formula, basically, which is inflated by correlation. So it'll have one part, which is the expectation of delta P squared over 1 generation, which is the classical thing with no correlations, plus the extra bit. And they're saying that basically the rate of drift, the rate of allele frequency drift, is inflated by 20% on average. The black curve is the average over all the replicates. These different colored curves are the various replicates. And actually the colors correspond to these different colors, I think. So actually that sort of shows that there's a substantial fraction of variance and therefore an inflation in the rate of drift due to linked selection. Interestingly, I don't think they come up with an explicit, or at least not a very confident explicit estimate of additive variance in fitness from this. So I think there's still quite a bit of work to do to really know how to analyze these sort of time series data. And as you can see, it's quite noisy, even with genome-wide data, with quite a few replicates, etc. It's not so easy to do, and it will be even harder in the natural population, I think. Though no doubt Daphne are the ideal situation. Okay, I think I'll stop there because what I'm going to go on to next is stabilizing selection. Well, it's a whole topic in itself. So. Which one? This covariance? No, this is the straight covariance in scale. So this is 10 to the minus 3, 2 times 10 to the minus 3. So this would mean that if we think of this point as the variance, then that's 2 times 10 to the minus 3. So the square root of that, well, that'll be 5. What is it? 0.05 change of allele frequency, something like that. Standard deviation. So it's a reasonable. So we're talking about smallish fluctuations of allele frequency. This is just covariance of delta P1, delta P2 of 2 adjacent or neighboring allele frequency changes. Okay, that's it. Any questions? Does anyone believe, maybe there's some real true Darwinians who believe there are no limits to selection? No. If you found a way to get beyond the limits, you could make a fortune in animal breeding. Okay, so the last session tomorrow morning, when we're all bright and sharp, so I can go faster, is stabilizing selection.
I was just curious about this result you showed about accumulation of information, how it relates to previous work you had done with Harold de Vladar on entropy, evolution maximizing entropy. Is this related in any way?
Because entropy thing, it sort of might be. That was something kind of. Yeah, I don't think there's a direct relationship. So that was work which we continue actually with Gaspar and Kabotova and so on in a number of papers. So then the idea is that you have a complicated sort of population genetic model, and it's got lots of variables, it takes a lot of computing power. How do you simplify it? And what you do is say that, well, all we care about are certain statistics, which might be the mean and the variance of some quantitative trait. So we don't want to deal with the genetic details. And what we do is assume that for given values of the observables, the mean and the variance of the trait or the distribution of the trait, the underlying allele frequencies are as random as possible in the sense of maximizing entropy. So they're as uniform as possible given the constraints. And it turns out you can then write down quite a neat set of recursions which give you the dynamics simply of the observable. So you might reduce a thing with thousands of allele frequencies to basically the means, the variance, or a small number of variables. And it works, it's extremely accurate. And we never worked out why it was so accurate. I mean, it's one of these things like the diffusion, which often is more accurate than it should be. So that's all good. In practice, calculating the recursions, even for simple models, is a real pain. And so it's just easier to simulate. But there's something intriguing there. But I don't think that there's anything directly connected with this information bound, which is actually a much simpler result. I mean, I was trying for years and years to find some bound, because you have the intuition that given a certain number of individuals, given a certain variance in reproductive success, there should be a limit to how far you can evolve a population in some sense. And so Michaud really got to this by just thinking of bounds on information. There are a series of nested bounds which say that basically the variance in fitness is not the fundamental quantity, but the fundamental quantity is the information introduced by some individuals living and some dying. Right? So there's an information measure at the level of individual reproduction, which you can propagate into a series of bounds on the way the whole population evolves. Yes, but it was information in a different sense. It was Fisherian information. So anyway, yeah, there's a whole zoo of ideas around this, some of which... Zoo is the correct word. Yes, yes. Yes, yes. Yeah. Well, I mean, in his way of thinking, the asymptotic rate would be Vm times the selection gradient, something like that. But of course, then you get to limits of what you can actually achieve. But if you think about it, I mean, animal breeders have very little ambition. I mean, you should be able to select a mammal, an arbitrary mammal, into a shrew or into an elephant. And that's happened. There's a chain of perfectly fit ancestors, or into a dinosaur, or I don't know. It's actually quite a tricky thought experiment saying how far could you take selection. Of course, you can't just take the genome, as in Jurassic Park, of a dinosaur and put it into a mammal and expect it to work. So you've somehow got to have the cytoplasmic machinery co-evolving with the genome, which is a bit tricky, but still gradual selection should be able to achieve everything that has actually been achieved by evolution.
Sorry.
Except, would you actually, maybe I had a question for you after your talk. Given that what we think of as the advanced complex organisms have simply become encrusted with all kinds of unnecessary complications, which are very hard to get rid of. Does that mean that we can't go backwards? I don't know.
I hear what you're saying. You should be able to take forward step by step. Look at things like
Yeah. Yes. But still, I mean, given enough time, you can evolve duplicate genes and they can have new functions and so on. So, I suppose it's a multi-year, multi-millennia NIH grant is to take a cave fish and select it back to a proper fish. Yes. No. Yeah.
Look, I think maybe you can explain me what do I really learn about our populations from this analysis?
I think. Yeah. So if you look at the marker allele frequency, 20% of that variance is due to actual selection, not just random drift. So it's bounding the total amount of selection that's going on. And one ought to be able to relate that to the amount of adaptation you can achieve. The trouble is you don't have a sort of measurable trait which tells you how much adaptation there's been. Maybe in an experiment where you're selecting, as you've got experiments at the moment going on, well, I think NADA has, where you're selecting on a known trait, then you might be able to ask how much of that can be attributed to sort of background stuff in the genome and how much is accounted for by major loci. I think that's the kind of question that interests me in these experiments. Can we partition the actual response into the various causes, small effects and big effects? And this is putting some sort of bound on the total response.
So the power means that it has reached this already.
No, the plateau only means that the correlations of allele frequency have decayed, and that's just a question of genome size, I think. So you've got a certain rate of decay of the whole genome, which Drosophila's got a pathetically small genome, so that's actually not so much. But in most organisms, you'd have a lot of stuff completely unlinked, and that would decay by half every generation. So really fast decay. And then you've got stuff on the same chromosome, you've got stuff very close. And so this is saying that there's a contribution from things of order, I suppose, 50 generations worth, that's 2 centimorgans. So within a couple of centimorgans, you've got stuff that's generating these correlations. Okay. So we've reached the limits to selection.